Meet Negaverse. It generates high-quality hard negatives, so AI learns real biology, not shortcuts. Every second, about 1 billion biochemical reactions happen inside a single human cell. Among them are protein-protein interactions, which control nearly every cellular process, how we walk, speak, and think. When they fail, diseases like cancer and neurodegeneration emerge. Yet after decades of research, we've mapped only 10 to 20% of all protein interactions. Discovering new ones experimentally is slow, often taking decades to reach a drug. AI could dramatically accelerate this, but only if it's trained on the right data. AI learns from positive and negative examples, but most negatives come from randomly pairing proteins, and not yet observed doesn't mean non-interacting. Many are actually undiscovered positives, teaching the model the wrong biology. Negaverse fixes this. Instead of random sampling or topology alone, it combines calibrated biological rules, biophysical constraints, network topology, and LLM-guided reasoning into one explainable framework, producing hard but honest negatives without hidden false negatives. Starting from your positives, Negaverse generates candidate non-pairs, then screens out known positives with biogrid, int-act, and string. The rest are scored in parallel, topology, structure, and biophysics, and the most ambiguous go to an LLM for literature reasoning. So what makes a good hard negative? Challenging enough that the model learns real biology, but not so close to positives that it becomes a hidden false negative. On the interactive map, real interactions, random pairs are chosen negatives, and the risky tails separate on network shape, biology, and chemistry. Hover any risky pair, and an LLM literature verdict explains it. Against random negatives, Negaverse cuts leakage from 7.6% to under 1, over 10 times cleaner. Here's Negaverse in action. One command screens the pool, places every pair on biological axes, escalates the ambiguous ones to literature review, and writes a live, interactive report, leakage under 1%, thousands of clean hard negatives. Each dataset, WHO, Rye, Dryad, and SARS-CoV-2, becomes a map you can rotate and hover for the model's reasoning. And this could go further. Trained on Negaverse negatives, a model like Descript already generalizes better than random to unseen proteins, proposing novel candidates that AlphaFold could then triage. Still experimental, but promising. Today, protein-protein. Next, protein-ligand, RNA protein, and more. Negaverse plausible from impossible. Thank you.